Chapter 8, Venipuncture Equipment. Our phlebotomy equipment is going to include our needles, the tubes that we collect the patient's blood in, plus the materials to ensure that the vein can be located, make sure that the puncture site is clean and the sample is labeled and transported correctly. Phlebotomists use a portable tray to carry all their necessary equipment in. We need to make sure that we keep our portable tray well stocked, clean, and organized. To clean our area, we would use a 10% bleach solution. That's going to work very well to disinfect. We are going to encounter patients in two different settings, inpatient and outpatient. We may also have an area that we have stocked that's our, instead of carrying that portable tray, maybe we're stationary in the lab and we're working from a stationary spot. Locating veins, we've worked on this in class. The tourniquet is going to be the most common way to locate a vein. The tourniquet is going to prevent venous blood flow out of the arm, causing those veins to bulge. The tourniquets may be latex or non-latex, so remembering allergies is very important. Once a tourniquet has been used, it should be disposed of to reduce the risk of pathogen transmission between patients. When cleaning and protecting that puncture site, the most commonly used antiseptic is 70% isopropyl alcohol or rubbing alcohol. Isopropyl alcohol is a bacteriostatic, which means that it inhibits the growth of reproduction of bacteria but does not kill them. Apply a 2x2 inch gauze square to stop bleeding to the puncture site after the needle removal. Our features of needles, all the needles have a point, a bevel, and a shaft. So the point is that sharp spot that provides smooth entry into the skin with minimum amount of pain. The bevel is that angle, that opening in the needle eases the shaft into the skin. And then the shafts differ in both lengths and gauges. The gauge is going to describe the diameter of the needle's lumen. The larger the number, the smaller the lumen. The choice of gauge is not a matter of personal preference. It depends on the type of collection and the condition of the patient. The hub is where the needle attaches to the collecting tube or the syringe. Multi-sample needles, while one tip of the needle penetrates the patient's skin, the second tip pierces the rubber cap of an evacuated collection tube. The retractable rubber sleeve covers the second tip when not inserted into the tube. Safety syringes and safety syringe needles. By slowly drawing back the plunger, a syringe provides that controlled, gentle vacuum for those fragile veins, like we've talked about in class. A syringe needle, also called a hypodermic needle, fits onto the syringe barrel. Winged infusion sets or butterflies, we use those for venipuncture on small veins, such as those in the hand and in elderly or pediatric patients. The most common needle size is a 23 gauge and 3/4 inch long. Most butterfly needles are now designed to be used with an evacuated tube system. Needle safety, OSHA issued a directive in November of 2000 stressing the use of safety devices to help reduce the number of sharp injuries. The Needle Safety and Prevention Act specified the different types of engineering controls that mentioned in OSHA's bloodborne pathogen standard to increase needle safety. Needle adapters. For most collections, you are going to use a multi-sample needle and an evacuated collection tube. We need to ensure a firm, stable connection between these two essential parts. A needle adapter, also called the tube holder, is used. Adapters have a tube advancement mark indicating how far the tube can be pushed in without losing the vacuum. Evacuated collection tubes. These are going to be color-coded. Which tube to use is going to depend on what tests have been ordered. Each type of tube may contain different sets of additives. The additives are going to be those chemicals designed to promote or prevent certain changes in the blood sample. Different types of blood specimens. We have whole blood. Whole blood is mixed with an anticoagulant so that it will not clot. This is for the hematology cell counts. The serum is the fluid portion of blood that remains after clotting. Plasma is the fluid portion of blood, including fibrinogen and other clotting factors. Any tube containing an additive must be inverted and mixed well immediately after removal from the adapter. Most need 5 to 8 inversions. Tube additives are identified by the stopper color, and then many tubes are coated with silicone to prevent blood from adhering to the wall and to slow the clotting. Anticoagulants are what prevent blood from clotting. Different anticoagulants work in different ways to prevent clotting and yield plasma. The EDTA, the citrate, and the oxalate bind to calcium, and the heparin inhibits conversion of prothrombin to thrombin. The choice of anticoagulant is going to be determined by the test to be done. Clot activators, thrombin directly increases clotting, and it is used for stat serum chemistry determinations, or if the patient is on anticoagulants. Clot activators may be adhered to the side of the tube, so therefore the sample must be inverted 5 times to allow the blood to come in contact with that activator. Polymer gel, an inert synthetic substance where the density is between that of cells of blood serum or plasma. When the specimen is centrifuged, the gel becomes a liquid and moves between the lower cell layer and the upper serum or plasma layer. So color-coded tops, the tube, if it's yellow, it's sterile. The additive is going to be sodium polyanthiosulfonate, or SPS, which is going to inhibit complement and phagocytosis. The specimen we're collecting in that is a whole blood. And the tests, we use this tube to recover microorganisms causing a blood infection. Our light blue tubes, so the tests are coagulation tests, prothrombin time. The additive in those is sodium citrate, which is an anticoagulant, and then the specimen is going to be plasma. Our red tubes come in either plastic or glass. The tests are going to be chemistry, serology, or blood bank. Our additives are clot activators in the plastic, and there's none in the glass. Our specimen is serum. Our lavender tubes, the test that we see ordered for these tubes is complete blood count of the CBC, sedimentation rate, routine immunohematology. Our additives are the EDTA, which again is an anticoagulant. Our specimen is whole blood. Our white top tubes, the tests are viral loads. Our additives are the polymer gel with EDTA, and the specimen is plasma. Our gold BD Hemoguard closure or red-gray tubes, these are going to be most chemistry tests. The additive is a clot activator or a polymer gel, and our specimen is serum. Our gray tubes, we test for lactic acid measurement, glucose tolerance test, our fasting blood sugars, our blood alcohol levels. The additives in these are anti-glycolytic agent, which preserves glucose, and then perhaps the anticoagulant potassium oxalate or heparin, and our specimen is plasma. Black tubes, which I have rarely seen, but these tests are going to be sedimentation rates. The additive is buffered sodium citrate, and our specimen is whole blood. Our green tube, the tests are stat and routine chemistry tests, ammonia levels, electrolytes, arterial blood gases, or ABGs. Our additive is sodium heparin, lithium heparin, or ammonium heparin, and our specimen is plasma. Our light green or greenish gray, our tests are stat and routine chemistry tests. Our additive is heparin, polymer gel, and our specimen is plasma. Orange BD Hemoguard closure or yellow gray tops, again stat chemistry. Our additive is thrombin, and our specimen is serum. Royal blue tubes, these are our toxicology, trace metals, nutritional analysis tests. Our additives are heparin, EDTA, and sometimes none, and our specimen is plasma or serum. Our tan top tubes, these are lead analysis. Our additive is heparin, and our specimen is plasma. Our yellow non-sterile, the tests are human leukocyte antigen, or the HLA studies. The additive is acid citrate or dextrose, and our specimen is whole blood. Pink top tubes, these tests are blood bank compatibility tests. Our additive is K2 EDTA, and our specimen is whole blood or plasma. Order of draw. So blood culture tubes, if we need to draw them, are always going to be drawn first. This prevents the transfer of unsterilized material from other tubes into that sterile tube. Second would be our light blue tubes. These tubes are always drawn before tubes containing other kinds of anticoagulants or clot activators, because the other additives could again contaminate this tube and interfere with coagulation testing. This blue tube is also the tube that has a fill level where it has to be filled to a certain level or they cannot use it. Glass red top tubes or plastic red top tubes may be drawn now. And then our red, gray, or gold BD HemoGuard tubes and plastic gold top tubes are next. We're going to see the gold tubes more than we're going to see that red-gray tube. Next then would be our green or our light green tubes, because the heparin is less likely to interfere with the EDTA tubes. And normally they say between the two greens, there's a darker green and a light green tube. Those can be interchanged, so it doesn't matter which one you draw first. And then next would be our lavender tubes. The gray top tube is last. This tube contains potassium oxalate. The potassium would elevate the potassium levels measured in the electrolyte analysis, and the oxalate can damage cell membranes, so that's why we would want to draw that gray tube last. Needle disposal containers, so we want to make sure that our needles are disposed of in the appropriate container, which is our sharps containers. They are red. I know you guys are all familiar with those. A used needle is a biohazard waste, and it must be treated as such. So to dispose of the needle with the adapter still attached immediately after activating the needle safety device, the needles must be placed in a clearly marked puncture-resistant biohazard disposable container. If you have any questions, please write them down and send them to me in an email, and I will either answer them by email or I will answer them for you next week when I'm back in class.